If you wanted to understand exactly how the security system in a, massive high-tech bank worked, you probably wouldn't learn much by just asking the bank manager.
Right, because they'd just give you the sanitized surface-level PR version.
Exactly. But if you really wanted to know every hidden wire, every blind spot, and every single pressure point, you would ask the most successful bank robber in history.
Because the robber has spent their entire existence figuring out exactly how to break that specific system.
Yeah, and today, for all of you listening, we are taking that exact approach, but to human biology. Welcome to today's deep dive. We are unpacking a truly monumental 2026 paper from the journal Cell.
It's titled Systematic Discovery of Pathogen Effector Functions Across Human Pathogens and Pathways.
Right, and instead of just studying human cells in isolation, these researchers decided to interrogate the greatest cellular bank robbers on Earth, which are viruses, bacteria, and parasites.
It is such a brilliant shift in perspective. Pathogens have spent millions of years running the ultimate, brutal clinical trials on our biology.
Which is a crazy way to think about it.
It really is. The researchers realized that if they could isolate the specific tools, like the lockpicks and crowbars, that these pathogens use, they wouldn't just learn about infections, they would create a master blueprint of the human cell itself.
Which brings us to the core of this paper. The researchers built something called the eORFeome. It's this massive library, but before we get into what's inside it, can you break down that term? What exactly is an eORFeome?
Yeah, so let's start with the base term ORF, which stands for open reading frame. In genetics, an open reading frame is essentially a stretch of DNA or RNA that can be read by a cell and translated into a protein.
So it's basically the instruction manual for a single biological machine.
Exactly, and the E stands for Effector. So the eORFeome is a library of open reading frames that specifically code for effector proteins.
And effectors are the weapons, right?
Right. Effectors are the specialized proteins that pathogens inject or produce inside a host cell to hijack it. And this specific library contains 3,835 of them. Yeah, we're talking about proteins drawn from a massive variety of enemies. Every major class of virus is represented, along with effectors from bacteria like Legionella and Chlamydia.
The ones that physically inject their proteins directly into our cytoplasm.
You got it.
Okay, so they've essentially collected nearly 4,000 different alien keys. But how do you actually test that many keys to see what cellular doors they open? I mean, you can't just dump them all into a petri dish of human cells at once, right? It would be total chaos.
Oh, it would be completely impossible to parse. The testing mechanism they designed is really what makes this paper a landmark. They used a barcoded lentiviral library.
Okay, a lentivirus. That's a type of virus that can permanently insert its genetic material into a host cell's DNA, right?
Yes. But here, they've stripped out all the dangerous, disease-causing parts of the lentivirus. They're using it strictly as a delivery truck.
Oh, okay.
Each viral truck is loaded with the genetic code for just one specific alien key, one eORF. And to keep track of everything, they attach a unique genetic barcode to each effector.
Sort of like scanning a label onto a package.
Exactly. So you have a fleet of over 3,800 different delivery trucks.
And then they just unleash them.
They infect millions of human cells in a massive batch, but they do it at a very low multiplicity of infection, or MOI.
Wait, MOI, I've heard that term, but what does it actually mean in practice here?
MOI is simply the ratio of infectious agents to target cells. By keeping the MOI very low, say 1 delivery truck for every 10 human cells, they ensure that the vast majority of cells don't get infected at all.
Oh, I see.
But the ones that do get infected almost certainly only get hit by one single delivery truck.
Okay, so you don't have a situation where one human cell accidentally gets 5 different keys and you have no idea which one did what. Each cell gets exactly one and only one pathogen effector.
Precisely. Now here is the really clever part. They didn't want the keys being manufactured right away. They used a doxycycline-inducible system.
Doxycycline? Wait, isn't that an antibiotic?
It is, yeah. But here it's used as a chemical switch.
Yeah.
The lentivirus inserts the code for the effector into the human cell's DNA, but it's engineered to be totally silent.
So the human cell just sits there, completely normal, holding this foreign blueprint.
Right. It's not until the researchers physically add doxycycline to the environment that the switch flips.
That is wild. So you add the switch, and suddenly, completely simultaneously, thousands of different human cells start manufacturing thousands of different alien weapons.
In one fell swoop. Then they wait a specific amount of time and use a technique called FACS, fluorescence-activated cell sorting, to physically sort the millions of cells based on what happened to them.
Like, did the cell die or did it survive a toxic chemical?
Exactly. Or did it turn on a specific glowing fluorescent reporter they built into an immune pathway?
And once they sort them into buckets, like these cells turned on the alarm and these cells silenced the alarm, they just read the barcodes.
You got it. They sequence the barcodes in each bucket, and instantly they have a master list of exactly which pathogen protein caused which specific reaction in the human cell.
Okay, so let's look at what this massive sorting operation actually found. And naturally, if you're a pathogen breaking into a cell, the absolute first thing you have to do is cut the cellular alarm system. What is the primary alarm they looked at?
They looked at the NF-kappa B pathway. NF-kappa B is a protein complex that is arguably the master regulator of host immunity and inflammation. It is the central alarm bell.
How does the bell normally get rung?
Well, when a cell senses danger, say, it detects a chemical called TNF-alpha in its environment.
Which is like a big red flag sent out by neighboring infected cells.
Right. It triggers a cascade. This cascade is meant to free the NF-kappa B complex so it can travel into the cell's nucleus, bind to the DNA, and turn on hundreds of genes designed to fight the infection.
So a pathogen desperately needs to stop NF-kappa B from getting to the nucleus.
And the screen found a massive toolkit designed to do just that. They identified 29 activators and 42 inhibitors of this pathway.
Wow. Did they find any famous ones that we already knew about?
They did. The screen successfully flagged the E6 protein from HPV and a salmonella effector called GTGA, which literally acts like molecular scissors to chop up one of the NF-kappa B subunits.
Okay, well, it's good that the screen found the known ones. It proves the system actually works. But the paper highlighted a completely novel discovery here, right? An effector from Legionella pneumophila, the bacteria that causes Legionnaire's disease.
Yes, an effector called RAVE, or LPG0195. Legionella is a fascinating bacteria. It lives inside our cells and uses a syringe-like mechanism called a type V secretion system
to inject hundreds of different proteins into the host cytoplasm.
Exactly. But until this eORFeome screen, nobody had a clue what RAVE actually did.
Okay, so the screen shows that RAVE stops the NF-kappa B alarm. But how? If other viruses use molecular scissors, what is RAVE doing?
To understand that, you have to know how the alarm is kept quiet in a healthy cell. Normally, NF-kappa B is held inactive in the cytoplasm by a brake protein called I-kappa B alpha.
So it's basically parked with the emergency brake on.
Right. And when a danger signal like TNF-alpha comes in, the cell's first move is to destroy that brake. Once the brake is gone, NF-kappa B is free to rush to the nucleus.
So the cell destroys its own brake to hit the gas.
Exactly. But what the researchers found is that RAVE prevents the cell from destroying the brake.
So it's not chopping up the alarm itself. It's more like taking a bottle of super glue and gluing the emergency brake onto the engine.
That is a great way to visualize it.
Even when the cell is desperately trying to hit the gas because it sees TNF-alpha, the engine won't turn over. The brake just won't come off.
Right. But the researchers wanted to know the exact molecular target RAVE was superglowing. To find out, they used whole cell mass spectrometry to essentially weigh and identify all the interacting proteins.
And what did they find?
They discovered that RAVE specifically targets and downregulates a critical host protein called RIPK1.
RIPK1, what's its job?
It's an enzyme, a kinase, that helps transmit that initial danger signal from the surface of the cell down to the brake. Without RIPK1, the signal never reaches the brake, and the brake never gets destroyed.
This is where the paper goes incredibly deep into structural biology, which I found fascinating. They didn't just stop at identifying RIPK1, right?
No, they wanted to know how RAVE's physical shape allowed it to do this. So they used an advanced AI tool called SoldSeq to predict RAVE's 3D structure.
Oh, wow. AI structural prediction. What did it look like?
They discovered that its shape closely resembles proteins in something called the TK superfamily.
TKI, like a tiki torch.
It's spelled the same way, but it's a family of enzymes. Interestingly, this family includes a known plant pathogen effector called HPA1.
Okay, so by aligning the 3D structures of RAVE and these other enzymes,
they pinpointed the likely business end of the RAVE weapon. They suspected 2 specific amino acids were doing the actual chemical work: a glutamic acid at position 19 and a histidine at position 21.
E19 and H21.
Exactly. To prove it, they mutated just those 2 specific residues, changing them to a neutral amino acid called alanine. And when they tested this mutated RAVE, it was completely broken.
It couldn't stop NF-kappa B anymore.
Not at all.
Wow. Okay, so we're about 5 minutes in. Let's just do a quick recap for you listening to make sure we're tracking this. We've got this massive eORFeome screen of almost 4,000 proteins.
Right.
And NF-kappa B is our cellular alarm. Legionella injects this totally unknown protein, RAVI, which acts like superglue on the alarm's break by targeting a host messenger called RIPK1.
And the window is so precise that changing just 2 single amino acids, E19 and H21, completely breaks the weapon.
Exactly. So here is an interactive question for you all at home to chew on. If you were designing a drug to stop runaway inflammation, how could mimicking RAVI's specific attack on RIPK1 give you a blueprint? Just something to think about.
It's a huge potential therapeutic avenue.
Okay, so the pathogen has successfully glued the alarm. The primary immune response is muted. Does the cell just sit there and accept its fate?
Not at all. The cell is highly resilient. If it realizes the primary alarm isn't working and it senses that its DNA is taking damage, it moves to plan B.
Which is what?
It attempts to lock the building down or initiate a self-destruct sequence to stop the pathogen from replicating. It does this using a protein called P53.
Ah, P53, the famous guardian of the genome. I know this one is a major tumor suppressor.
It is. When P53 is activated by cellular stress, it binds to the cell's DNA and turns on specific genes like P21, which physically halts the cell cycle.
And if the damage is too bad, it triggers apoptosis, right? Programmed cell suicide.
Exactly. Naturally, viruses have to counter this, or their host factory will just shut itself down.
Right, and I know high-risk HPV handles this by just outright destroying P53, but the eORFeome screen pulled up a totally different strategy from a virus called human herpes virus 6A, or HHV6A.
Yes, a protein called U14. And this is where we see the incredible, terrifying diversity of viral evolution. You'd assume U14 just shreds P53 like HPV does, right?
Yeah, I mean, that seems like the easiest way to deal with the security guard. Just take him out entirely.
But U14 doesn't destroy P53. It sequesters it.
Sequesters it, like puts it in a cage.
Physically, yes. U14 forces P53 into tight, abnormal clusters within the cell's nucleus, called nuclear foci.
So instead of throwing the security guard out the window, U14 just locks him in the bathroom.
That is exactly what is happening.
He's technically still alive, still in the building, but he is completely incapable of doing his job.
Because P53 is trapped in these bathroom clusters, it physically cannot reach the DNA to bind to it. It can't turn on P21, the cell cycle continues, and the virus keeps replicating.
That is so smart.
And there are 2 massive evolutionary twists to this U14 story. First, HHV6A has a very close viral cousin, HHV6B.
Okay.
They are remarkably similar viruses. Their U14 proteins are almost identical. Yet the HHV6B version of the protein does not bind P53 at all.
Wait, really? 2 closely related viruses, but one has the bathroom lock and the other doesn't.
It shows how incredibly fast and specific these evolutionary arms races are. A tiny shift in genetic code completely changes the survival strategy.
Wow. What's the second twist?
If you look at the physical structure of how HHV6A's U14 binds to P53, it perfectly mimics the binding interface of a completely unrelated viral protein, the SV40 large T antigen.
SV40, wait, that's convergent evolution.
Exactly. 2 completely different viruses, evolving in entirely different environments, completely independent of one another, somehow arrived at the exact same molecular blueprint to lock the P53 security guard in the bathroom.
That implies that this specific binding pocket on P53 is like a fundamental universal weak point in our biology.
It does. And to prove how devastatingly effective this sequestration tactic is, the researchers tested U14 in an in vivo mouse model. Specifically, they used FSF G12D Rosa 26 crash 2 mice.
Whoa, alphabet soup alert. Stop right there. For me and you listening, we don't need to know the passwords. We just need to know what these mice actually prove. Why use them?
Fair enough. Put simply, these mice were genetically engineered to have a deadly, highly aggressive lung cancer gene waiting in their DNA. But that cancer gene is trapped behind a genetic locked door or a stop sign. It's completely dormant.
Okay, so they have a ticking time bomb, but the timer is paused.
Right. The researchers can unpause the timer by using a lentivirus to deliver a specific protein into the mice's lungs that removes the stop sign.
And normally when you open that door and the cancer starts trying to grow, the mouse's P53 guardian wakes up, senses the runaway replication, and fights back hard, right? Suppressing the tumor.
Exactly. But they delivered the U14 protein along with the cancer trigger.
Ah.
In one group of mice, they opened the cancer door and simultaneously delivered U14 to lock the P53 guardian in the bathroom. And the results were stark. Delivering U14 accelerated the lung tumors just as effectively as if they had gone into the mouse's DNA and completely deleted the p53 gene entirely.
That is staggering. Okay, let's do a quick recap of Section 2 for everyone. p53 is the cell's guardian. And we just learned HHV6A's U14 protein doesn't destroy the guardian, it just locks it away in nuclear foci.
Mimicking SV40 in a perfect example of convergent evolution.
Right, and it's so powerful it drives explosive lung tumors in mice just by keeping the guard occupied. So for you listening, here is our second interactive question. Knowing that HHV6A U14 evolved this locking mechanism entirely independently of other viruses, what does this tell you about the ultimate weak spots in our cellular armor?
It really makes you wonder how many other viruses have found that exact same backdoor.
Absolutely. Okay, so the main alarm is superglued by Ravi. The p53 lockdown is trapped in the bathroom by U14. What is the cell's plan C? It has to have a final move.
Its final move is to desperately try to wave a flag out the window, hoping the systemic immune system, the P cells patrolling outside, will notice something is wrong.
And the researchers looked at two interconnected pathways here, right? STING and MHC-I. I know MHC-I is the flag on the surface, but what is SDM?
STING stands for stimulator of interferon genes. It acts as an internal tripwire for the innate immune system.
How does it get tripped?
Normally, DNA is kept safely inside the nucleus. But if a virus is replicating in the main body of the cell, the cytoplasm, it often leaves loose bits of viral DNA floating around.
Which definitely shouldn't be there.
Exactly. The cell has sensors that detect that loose DNA and activate STING. STING then produces a messenger molecule called cGAM, which triggers massive inflammation and often cell death. It literally burns the factory down to stop the virus.
And MHC-I.
MHC-I, or Major Histocompatibility Complex Class I, is the adaptive immune system's flag. It constantly takes little chunks of proteins from inside the cell, including viral proteins if the cell is infected, and displays them on the cell surface.
So it's literally holding up a sign to passing T cells saying, hey, look what's inside me, I'm infected, destroy me.
That's exactly it. So a smart virus would want to silence the STING tripwire so the factory doesn't burn down, and take down the MHC-I flag so the T cells don't attack.
Usually a virus has a totally different tool for each job, right?
Usually, yes. But the screen found a viral protein that pulls off an incredible double-duty act to stop both of these processes simultaneously. It's called U21, from human herpes virus 7, or HHV7.
Now wait, wasn't U21 already known to scientists? I feel like I've heard of it.
It was. We already knew U21 was bad news for MHC-I. We knew that U21 grabs those MHC-I flags before they reach the surface and routes them to the lysosome.
Which is essentially the cellular trash can or incinerator, right?
Exactly. So it throws the flag in the incinerator. But the eORFeome screen revealed a completely hidden second function. U21 also potently degrades STING.
No.
Yes, it intercepts the STING pathway and throws that into the lysosomal incinerator too.
So U21 is like a cellular bouncer with two hands. With one hand, it grabs the MHC-I flag. With the other hand, it grabs the STING DNA sensor, and it just walks them both down the hall and throws them into the incinerator at the exact same time.
That's a perfect visual. But the researchers wanted to know how one single protein could juggle two totally different biological targets. So they looked at U21's structure, specifically focusing on its C-terminal tail.
The C-terminal tail.
It's the very end of the protein chain that anchors it into cellular membranes. They found two specific hydrophobic motifs, essentially little oily patches in this tail sequence.
And they thought those patches were grabbing STING.
Yes. So they mutated them. They created specific alterations called the U21 NRD mutation and the U21 4D mutation.
And did mutating the tail break the bouncer?
It broke exactly half of the bouncer. When they tested the mutated U21, it could no longer destroy STING. The cell was able to sense the DNA and trigger its offenses.
Oh, wow.
But, and this is the fascinating part, the mutated U21 could still perfectly destroy MHC-I.
That is incredible. So they proved that U21 has geographically separate domains. You can literally snip the function of one hand off the bouncer without affecting the other hand's ability to throw out the flag.
It proves that U21 is a highly modular dual-function effector. It evolved two distinct tools on one physical frame to inhibit both the adaptive immune response and the innate immune response.
Okay, time for our third recap. For you listening, we've got fencing sensing loose DNA and MHC-I signaling for help. HHV7's U21 protein destroys both of them using different parts of its structure, and mutating the tail separates the functions.
The true multi-tool.
So here's a question for you. Imagine if we could snip off just the STING-inhibiting tail of U21. Could we use that fragment to treat autoimmune diseases where the body mistakenly attacks its own DNA?
It's exactly the kind of outside-the-box thinking this paper encourages. The efficiency of these viral tools is just terrifying. We're seeing how pathogens slowly adapt these intricate weapons over millions of years.
But here is where the story takes a truly mind-bending turn, right? Because it's one thing for a virus to slowly adapt a weapon. It's a completely different level of biological genius to invent a brand new weapon out of thin air.
Yes, and this brings us to one of the most massive finds from the MHC-I display screen, the 13.6K protein. It's also known as the I-leader protein, and it's found in specific strains of human adenoviruses, like A12 and F41.
Okay, I know adenoviruses cause things like the common cold or pink eye. What is this 13.6K protein doing?
It stops the MHC-I flag from working, but the way it evolved to do this is a genetic miracle. The gene for 13.6K evolved de novo, meaning entirely from scratch, specifically in primate adenoviruses.
Evolved from scratch. How?
It's the location of the gene that is so staggering. It is located on the exact opposite strand of a highly essential viral gene called the E2B polymerase.
Okay, wait, let me unpack this because I think I know why that's crazy. DNA is a double helix, right?
Yeah.
Two strands wrapped around each other like a twisted ladder. Usually a gene is read off one side of the ladder.
Right.
And the E2B polymerase on one side is absolutely essential for the virus to survive. It can't afford any typos there. Any mutation in E2B usually means death for the virus.
It is tightly, rigidly controlled code.
But you're saying that on the reverse side of that exact same tightly controlled stretch of DNA tape, the virus managed to evolve a completely new, perfectly functional gene reading the exact same letters backward.
Yes. Evolving overlapping genes without destroying the original essential function is almost impossibly difficult.
Wow.
Think about writing a brilliant, award-winning poem. Now read the letters of that poem backward and imagine it forms a completely different, perfectly coherent instruction manual for building a complex machine.
That is deeply unsettling. Nature's backward poetry. So what does this backward engineered machine actually do?
The 13.6K protein embeds itself into the membrane of the endoplasmic reticulum, or ER, which is a major manufacturing center in the cell. And it acts as a direct inhibitor of something called the TAP complex.
TAP transporter associated with antigen processing. What does TAP normally do?
Remember those chunks of viral proteins we said MHC-I displays on the surface?
Yeah, the cargo for the flag.
Those chunks have to physically get from the main cell body into the ER so they can be loaded onto the MHC-I flags. TP is the molecular pump that transports them inside.
So 13.6K binds directly to the T pump and jams it.
Exactly.
If the pump is jammed, no viral protein chunks get into the ER. The MHC-I flags are starved of cargo.
And if the flag is empty, it never gets sent to the cell surface, and the immune system remains completely blind to the adenovirus infection.
And the researchers proved this, didn't they?
They proved it beautifully using a fluorescently labeled peptide assay. They basically made glowing protein chunks that they could watch under a microscope.
Oh, that's clever.
Normally, you'd see those glowing chunks get pumped into the ER, but when they introduced the 13.6K protein, the glowing peptides were completely blocked. They couldn't enter. This established 13.6K as a completely novel, recently evolved family of viral TAP inhibitors.
Okay, time for our final section recap. Adenoviruses basically invented a new gene backward over an existing one. This 13.6K protein binds the TT transporter and stops peptides from reaching MHC-I.
A perfectly evolved jammer.
So here is our final interactive question for you to ponder. If a virus can invent a functional protein from the quote-unquote wrong side of a DNA strand, how much hidden biology is lurking in the unread parts of our own human genome?
That is the million-dollar question in genetics right now.
So let's zoom out and put this all together. We started with this massive library, this eORFeome of nearly 4,000 alien keys, and we've seen them superglue alarms with Ravi, lock tumor suppressors in the bathroom with U14, juggle multiple threats with the U21 bouncer, and literally write backward genetic code to jam the TAP pump with 13.6K.
It's not just a spreadsheet of viral parts. It is a collection of over 3,000 perfectly evolved biological tools.
The researchers referred to them as perturbogens, right?
Yes. They are ultra-precise keys that reveal the exact pressure points of human cells.
And that leads to a really profound thought. We humans spend billions of dollars and, decades of time in laboratories trying to design synthetic drugs from scratch that can precisely alter human biology to cure diseases.
We do. And we often fail because human biology is just incredibly complex. But the pathogens, They've already done the work. They've spent millions of years running trials, perfecting these tools to manipulate our pathways with absolute precision.
What if the next breakthrough cancer treatment or the cure for a devastating autoimmune disease where the body attacks its own DNA isn't designed by a supercomputer? What if we could just borrow from the eORFeome?
Exactly.
We could synthesize the super glue of rave to calm runaway inflammation, or snip off the tail of U21 to control a hyperactive immune system. We could rewire human cells for our own benefit using the tools our oldest enemies built.
It entirely shifts our perspective. Pathogens are no longer just the agents of disease. They might actually be the most advanced biochemical engineers on the planet, and with this paper, they've effectively just handed us their master toolkit.
It changes everything about how we look at an infection. We aren't just taking pictures of the murky waters of human biology anymore. We are finally finding the steering wheel. Thank you for joining us on this deep dive. For you listening at home, keep questioning the microscopic world around you because, as we've seen today, the ultimate cures to our biggest medical problems might already be hiding inside the viruses we fear the most.